The title of our project is Isolation and Screening of Probiotic Lactic Acid Bacterial Strains Having Inhibitory Effect Against Enterobacter cloacae. Introduction. Bacteriocins are ribosomally synthesized and post-translationally modified peptides. They have antibacterial activity against closely related sins. Their mode of action are pore formation, nucleus activity, and peptidoglycan inhibition. They have inhibitory effect against intestinal commensals. So in this study, we have evaluated the antimicrobial property of lactic acid bacterial strains against Enterobacter cloacae. So lactic acid bacteria, according to the World Health Organization, lactic acid bacteria are classified into probiotics, which are live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. So apart from bacteriocins, lactic acid bacteria also produce other low molecular weight substances. And these lactic acid bacteria are constituted in a diverse group of gram-positive microorganisms, which includes the lactobacilli, lactococci, streptococci, etc. The objective of our study is to evaluate the antibacterial activity of various bacterial strains which we have isolated from curd and batter samples against the indicated organisms. Next, we will be identifying the production of bacteriocins. And finally, we will be expressing the produced bacteriocin in a food grade expression system, and we will be increasing the yield of the protein production.
So this is our workflow. I will detailedly explain it later. Next method. So to isolate the lactic acid bacteria, we collected the curd and batter samples. Then we serially diluted the samples. To isolate the lactic acid bacteria alone, we did a spread and streak plate technique. The isolated colonies were pure isolated colonies were inoculated at 25 ml of MRS bath, then incubated at overnight 37 degrees Celsius. So to view the morphology of lactic acid bacteria, we did gram staining, then we view under the microscope. To check the antibacterial activity of the isolates, we did overlay SM method. First, we prepared the soft agar before the day of doing the bottom agar. The soft agar was prepared for 100 ml. The bottom agar was placed with isolated colonies at the center. Then it incubated 37 degrees Celsius. Then the soft agar was inoculated with the Enterobacter cloacae, which is an integrated organism. The soft agar was poured over the bottom agar. Then we incubated at the 37 degrees Celsius.
So here are the results. We got 8 isolates showing a zone of inhibition. The first isolate shown 4 mm of thickness zone of inhibition, and the second isolate shown 25 mm of zone of inhibition. Next, the isolate from batter sample shown 10 mm of zone of inhibition. Next, the curd sample isolate shown 2 mm of zone of inhibition. Then the isolate from batter sample shown 1 mm, and the next shown 2 mm of zone of inhibition. Finally, we got 15 mm of zone of inhibition showing by the curd sample isolates. The future work is we have to confirm the production of bacteriocin by doing neutralization by NaOH and inhibition of H2O2 and by adding proteinase K enzyme. So these are the references. Thank you.